Okay, so welcome everyone. Excited to see you all here. So this is the joint Broad and MIT EECS Colloquium, which is co-hosted by the Eric and Wendy Schmidt Center and AI&D within MIT EECS, where we're featuring speakers who really go both ways of working on very important biomedical questions and informing through that the types of machine learning problems that they're working on, and then bringing the machine learning developments back into the biomedical questions. And we'll see an amazing example of this here today with Anna Goldenberg. So I'm very excited to introduce to you Anna Goldenberg, who is a professor in the Department of Computer Science and the Department of Laboratory Medicine and Pathobiology at the University of Toronto. In addition, she's also the Varma Family Chair in Biomedical Informatics and AI at the SeaKids Research Institute, as well as CFAR AI Chair at the Vector Institute. So Anna got her PhD at CMU, where she was first working on graphical models and applications to social networks. And then during her PhD, started working on computational biology and more problems in medicine. And she was really one of the first ones to actually develop methods and to identify the need to develop methods that can really integrate very different data modalities in order to get new insights into different diseases, has worked at the beginning on data sets or data types or modalities like gene expression and methylation, try to integrate these while studying cancer. And the current focus of the lab is on developing and deploying ML models to healthcare, in particular in kinds of longitudinal and very complex longitudinal settings. And Anna's lab is really strongly committed to creating responsible AI to benefit patients across a variety of conditions. And so we'll hear a lot about the different time series types of models that Anna is developing in different kinds of disease settings. I'm very excited about it. So the floor is all yours, Anna. So thank you very much for coming to visit us.
Thank you very much for inviting me. This is my great pleasure to give a talk here and to tell you a little bit about the time series model journey. It started with one project, which I will mention in the beginning, in collaboration with a critical care unit at SeaKids Hospital, which is a children's hospital in Toronto, and evolved into this many different projects, not all machine learning, but all motivated by our desire to actually take the AI tools that we are building to benefit the patients. The motivating story is very simple. So this is what happens. We think of diseases and sick people and how we would like to help them. But the reality is that it can happen to anybody. And the story in ICU was that a 12-year-old boy was skiing, and he fell down and broke his femur. So he was delivered by helicopter to SeaKids, and they did a surgery, and the surgery was successful. But what happened was during the surgery, a clot has formed, and the boy suffered a cardiac arrest and unfortunately permanent damage to his brain. So this all happened within a matter of weeks. And this was happening at the exact same time as we had this project going about detecting and preventing cardiac arrest. And so it just highlighted how important some of this work is or can be for patients who are not even patients yet. So I don't know if you know the meager statistics of cardiac arrest. So at home, if it happens, the chance of survival is about 14%. At the hospital, where there are really strong teams and they know exactly what they're doing, they can do it really fast, the rate of survival is only 36%. So the only way, really, to make sure that nothing happens is the prevention. And it's not that people have not been trying to prevent it, right? So this is the picture of SeaKids ICU. This was a courtesy of Peter Lawson, who is now here in Boston Children's Hospital. This is a picture of our ICU, and you can see how many monitors there are. Every single red circle is highlighting a monitor that is currently in ICU, and there are more. I'm sure I've missed some. So every patient has a bunch of monitors attached to them. And so there has to be nurses and fellows and all the people who are around an ICU monitoring the data that is streaming at all times to make sure that nothing goes unnoticed, that the patient is taken care of at the right time. And Peter actually always compares it to sort of the throughput of Niagara Falls. So Niagara Falls is roughly 200,000 cubic feet per second, and the data in ICU is about 200,000 bytes per second. So there's really a comparable amount of data to water ratio here. So we have formed a team with the ICU Lawson Labs, and this is my PhD student, Sana Tonicaboni, who has studied on this project, which said, how do we predict cardiac arrest? So the data that we had is all the streaming data from the Philips monitors that were shown on the previous screen. You have heart rate, respiratory measures, some blood pressure measures, some oxygen measures, etc. All of this stream in real time. We had 259 instances of cardiac arrest. We had about 1149 ICU patients that did not suffer cardiac arrest. And of course, for controls, you can also take some time before the cardiac arrest happened. So 5 hours before cardiac arrest happened, we consider that time a pre-cardiac arrest time. So you can use that as a control as well. Also note that we treat any 2-hour window right before cardiac arrest as the cardiac arrest window. Now, we are predicting in smaller window chunks, and so some of this earlier, within 2 hours, some of these earlier chunks are likely not actually cardiac arrest chunks. So some of them will say that it's cardiac arrest, but it's not quite yet. So the idea for this, you will see the chronology of this. We started with a architecture that actually worked quite well. So these were the convolutions that were coupled with LSTM model that later that went to predict cardiac arrest and estimate actually the risk of cardiac arrest, right? It's just the probability is what we were estimating. Unfortunately, or fortunately, this is how the data is, period, is that there's a lot of missing data, a lot. In time series, a lot of missing data. So what we ended up doing, previously we were pre-processing, we were putting a low filter on it. We stopped doing all of that, and we basically started using masks, and then a separate layer that calibrates the data to not incorporate, to downweigh the missing periods of the data when we are doing the computation. So this seems to work quite well. So you can see here the AUC is fairly good. The precision is potentially not great, but for this kind of data, it's okay. You can see the very high high-risk cohort, ECMO cohort. They have largely very high risk. The short-stay cohort largely has the low risk as an estimated, but you can see that the tails are there, right? The short-stay also get predicted quite high risk sometimes, and even the very high-risk cohort also has very low risk. And so we were concerned, is this even clinically relevant or clinically useful? And we were talking to Peter about this, and he said, actually, I think what we would like to use this tool in the end for is deterioration, right? Building up to cardiac arrest. It's not really specifically the prediction for cardiac arrest that works. It's really the sort of the chance of something wrong happening. You're probably capturing other critical events. And what happens here also is that we have prevented some of them, and it would have happened, and you signal the arrest, but you don't have the proper label. So all of that is in the data, of course. So when we started thinking about what it would actually come to and what we should expect, and we did a back of the envelope computation, and we have about 100 cardiac arrests at our unit per year, which is good. That's 365 days, 24 hours a day. We have about 30 beds. And this system would be running and predicting, let's say, every minute, every 30 seconds, right? It can predict any time. So if we have even 1% error rate, that we would get about 30,000 false positives for that event. So what we started thinking about, to predict cardiac arrest, we actually might not want to use this tool, right? Everybody talks about alarm burnout. So we really started pivoting to making it a deterioration estimation tool. And I think this, when you start really imagining your tool working at the bedside, you start pivoting and shifting in your mind about what question are you trying to answer and how is it going to modify the clinical workflow, and will it? Because if it doesn't, then what's the point of implementing it in the end? So, as part of this work, we sort of started thinking, okay, so what else do clinicians want to see or need to see? And there was a brief survey. So this is a foray that was not very comfortable necessarily for my students, but we had Shalmoy, who was a postdoc here and now has a faculty position, and Melissa, who is a bioethicist, and also my colleague right now, or was my fellow at the time, to figure out what clinicians want to know. So we went and did a survey of the clinicians in ICU, and we found out very interesting things. But among them, they're saying that we want to know what triggered the alarm, right? We want some kind of support to what should we do to our decision making. And also, for every event, they want to know what has influenced the score. And they were saying, we are actually perfectly fine with the system not being completely accurate or making mistakes, as long as we understand where the system's biases are. So if we understand how the system functions, we will be able to work with a system that is not perfect, and that's fine with us. And in terms of temporal explanations, they were saying, we really wish to understand what preceded this prediction of high risk. What actually triggered it, and this would help us to translate information between members of the team as well, and as well as translate between shifts between different teams. So from there was born another project, which is sort of the explainability tool for time series. And this was joint work with David Duveneau and Kieran Campbell that was published at NeurIPS in 2020. So the question is, how do we explain the predicted risk? And here, this is the same setup. We have the exact same data, and we've arrived at an event, and we are trying to see what, in the past, at which point did it deviate from the actual, from what we would have expected. So the model output is probability of y, which is our estimate of risk, given all the data. And we break it up, the importance score, into two things. One is what would be the estimate given all the data up to the previous time step. So if we didn't see any of the data before at this given time step, what would be the difference between the two estimates? And the second one, the second component of the score comes from the fact that we have seen the data and we want to try to understand which subset of features at this time step is actually important. So the second part is, if we were to have added the set of features of the given time step, how would it change our prediction from the previous time step? So then our the importance score, which we call fit, is the difference between the two scores. And the way to think about it is that if the subset doesn't matter at all, then our Keil divergence between the current time step and the previous time step would be exactly the same. And so the importance would be 0, right? And if it's really, really important, and if it explains everything of the current time step, then it would be a highly positive score. So this is the sort of the way, and we have tested by removing the top 50 most important features and compared it to a whole bunch of course, feature importance is a very common kind of method that's been developed, and you know the Sheff values, and you know the deep lift, and you know probably have heard of a whole bunch of different approaches, and we have compared what happens if we rank the features according to all of these different methods, and we remove the top 50 features that are considered important by each of those methods. And our method has the biggest loss in accuracy if we remove our features, which indicates that the features that it finds are most important, more important than other methods find.
When you say removing the top 50 features, one way to say it is, listen, maybe there's 5 groups of features, and these top 50, if other methods are choosing highly independent features of each other, then removing those could still keep the signal. If a method is keeping all of them, then you will actually lose the signal by removing all 50. Did you look into the correlation between the features that are being kept? Do you see what I'm getting at, basically? I'm just thinking of this as a metric.
I think I do see what you think, and let me correct me if I'm wrong, but I think the point is that these features are most predictive of the outcome. It doesn't matter if they're independent or dependent. I mean, this is not the question we are trying to solve. We are not trying to find the features that are most uncorrelated with each other. There are simple ways to do that. This is really the saying, I want to make sure that the features that I am selecting and they're in this time step for this particular event are the ones that are most predictive of what happens in the future.
As a metric, why not just show overall accuracy?
We do show overall accuracy, but it's about subsets, all subsets of features, right? So I guess one of the issues with everything that we try to do after predicting cardiac arrest is that a lot of the questions came from the unlabeled space, right? Saying which features are important is sort of, it's not a task where you have the ground truth for real data. You can construct examples where it will obviously do very well, but a lot of them are sort of the unlabeled questions that we have embarked on. So evaluation is always a question. So please, I don't mind questions. So the next question we sort of tried to embark on, which was a very different question, and the question of representational learning is trying to understand what are we actually seeing in the data, right? So we have in one case, we can recognize an event as a clinical intervention because it's labeled. In another case, we have some event, it looks different from what the patient usually experiences, and we have no label for it. And most of the events in this data are like that. So what we are trying to figure out is what are the states that the patient is in, and then trying to characterize whether the states are critical or deteriorative or not. So basically trying to map it to a low representational state. And so the idea of this next project was trying to figure out in the unlabeled space, what do we know, what do we not know? What assumptions can we make? And the assumption that we made here was that the windows that are right next to each other are more likely to be in the same state, similar to each other, whereas windows that are far away from each other are likely to be different. So we have a contrastive loss here for this representational learning. And you might know that there are several contrastive loss type projects that have been published, papers that have been published around 2020, 2021. And so we had something to build on, but they didn't really work well for this data. And so what we basically did then, once we had this hypothesis, we have looked at the discriminator between the windows that are close to each other and the windows that are far from each other. The way that we have determined the windows is by non-stationarity, and I think I talked to some of you about this test, is that in principle, for a while, I've been kind of disappointed with the time series modeling, because that it's always a very fixed window. So you have a fixed window and you kind of move it forward, and this window has no relation to how the data actually behaves. So what we did here is we have determined this big non-stationarity windows from which we sampled our window, neighborhood from which we sampled our windows by using, in this particular case, augmented Dickey-Fuller test. There are other non-stationarity tests that you can use. But basically, the idea is simple. You have a neighborhood, and you are trying to figure out whether the new window is in this, can you grow this, this neighborhood further? And if you, by doing non-stationarity test, if you say, okay, my next window is not coming from this distribution that I have already observed, then you can't grow it, and this is your neighborhood. Yeah, and so you can sample then windows from within the stationarity window, and you're hoping that they will be of the same state. And this is basically the loss, the contrastive loss that we worked with. The one thing that that we also had to incorporate is the sort of the bias that the windows that are far away could actually be in the same state, right? If you think of seasonality of time series signal, they could also actually be in the same state. So we have incorporated this sort of weighting of the positive unlabeled learning paradigm that allows you to to make sure that the windows from the negative regions that you don't really know are negative to downweight some of them. Here you can see the representations that we have learned. So this is TNC, this is our approach, the temporal neighborhood coding approach, and the triplet loss and the CPC that have been published in the past. And you can see that in terms of representational embedding, our embedding is a lot sort of cleaner than some of the other embeddings on the same data. But this was sort of the simulated data, and on the real data, we actually had to adjust it yet some more to make sure that the windows. So basically, what happened was it was picking up non-stationarity too often. So the test was very sensitive, and so our neighborhoods tended to be very, very small, and so it was not useful to us. So we had to do a lot of sort of learning of the parameters of how sensitive the test could be. So Edison is an undergrad in my lab, and we have done a lot of sort of adjustments to the method to actually make it work on the real ICU data that I was talking to you about. That is our primary goal. And you can see that there is embedding, and also the embedding tended to grow. We tried to make it a bit non-parametric, where we could have a large number of embeddings, and the number of the dimensions would tend to grow. So we actually had to do the pruning for the real data as well. So you can see that every single kind of highlighted area, there is a dimension that captures that there is something going wrong, and there are some dimensions that say, well, it's just a peak, and other dimensions say, well, this peak is a bit different from this other peak. So I think in general, working directly with the time series data, if you're not trying to predict something, but trying to understand something about the trajectory and the similarity of patients, then the representational learning is actually a must. You really want to project it to the smaller space, because otherwise, to the lower dimensional space, because otherwise it becomes very unruly and just too much noise. Yeah.
Are the 0 to 5 labels in the top chart, the top graph, meaningful, or are those just...
No, it's just the 6 dimensions that we prune to. Yeah. I think we have tried to sort of discuss with the clinicians afterwards whether this makes some sense to them, but labeling this is not easy even for them. Manolis?
So there's many ways to sort of learn these 6 dimensions, if you wish. One way would be at the union of all the data sets independently. Another one would be so that the transitions between them are smoother. I'm curious if your method allows you to do the latter, not just the former.
Yeah, the smooth transition was key.
Beautiful, beautiful.
Yeah, that's why you can see, like, the whole point of the neighborhood is that it's a distribution. And so you really don't have the sort of the hard boundaries of the prefix windows.
Yeah. And now when you go back and ask, what are the dimensions that I would have learned without these temporal data? And basically, what are the new things? Is it that they're slightly shifted? Are there entirely new things that are coming up? I'm just curious if you have some insights. I understand that, Peter's question is, how do you interpret these 6 dimensions? And if you can't do that, maybe it's harder to sort of say how they're different from another 6 dimensions. But I'm curious if you have some insights to share.
I think in our case, the dimensions are secondary to the states. So if we could interpret the states themselves, right? Because even the states are discovered. So if we could interpret the states, we'd be happy. Forget dimensions. we are trying to understand anything and everything about this data. So I feel that the dimensions in my mind, in a technical space, we're just trying to select the minimal number of dimensions that would still give us the differentiable factor between the states that we care about. So they don't necessarily have to have meaning other than being the minimal set.
So in a lot of traditional machine learning, the minimal set of dimensions would be, of course, the favored approach. There's been a realization with a lot of this deep learning sort of that if you throw more parameters at the model, you give it the opportunity to encode additional meaningful states. Have you kind of experimented with this idea of, what if we gave it more parameters than we think it needs?
We did. And then we found that it was just learning slightly different dimensions. So the dimensions were very, very similar. So you give it 16 dimensions, you give it 20 dimensions, you give it 30 dimensions. There are dimensions that are different, but they're similar enough that they don't really add value. They're just overfitting to the data. So in our case, it was not valuable. That's why we started working with a back contracting to the minimal dimensions and pruning.
Yeah, some people would tell you do 10 times as many dimensions, maybe it'll become more meaningful. But we can talk about it.
I think there is an understanding. There's an approach in machine learning to saying all I care about is the prediction of the outcome, my loss, right? I'm just minimizing loss. I don't care about what it learns. I don't care about how it learns. I will throw the kitchen sink at it with all the parameters in it, and it will be the best possible approach. In this particular case, we don't understand the data. Nobody understands this data. And so for us to try to figure out how to meaningfully address the problem, either with a fit, with a sort of the explainable features for the clinicians, or for trying to generate data back from it, we have to anchor ourselves somewhere, right? This is not a prediction task. This is an unsupervised representational learning task. So we have to be grounded somewhere.
Sorry for the distraction.
No, it's okay. It's an important question. I hear you. Yeah, and so another interesting point I think was looking at the trajectory in the embedded space, looking at the trajectory over time, so how close you are. So this darker dots here on the plot are ones that are further away from arrest, and the lighter ones are closer to the arrest. And you can see that they're embedded in different parts of the space, which gives us some hope that the representations that we are learning are actually meaningful. And this brings me to my most recent and therefore most favorite model. And this comes from also done with the same student in collaboration with Dave Bly at Columbia. And the key here was, again, understanding that even with the representational learning that we have done before, we are treating the whole population as a cohort. And that it's all of our studies, especially in the wearable space, but in physiological space as well, highlight the heterogeneity of the patients and the difference between these sort of representations per patient. So in time series space, we have we have models that model on the individual level. And the problem with those models is that you can't actually make the generalization to other patients when you see new patients, right? You have a model for that patient, but that patient is gone from ICU now. So what do you do with a new patient? Or you have models that are population-based that say, I have this sufficiently general model that works for the whole population. And that's also not entirely, fair to the data, because the reality is that people come with different kind of problems to ICU. There are new policies in place. There are new tools and technology. Everything evolves in medicine, thankfully, all the time, right? We're providing better and better care. So we can't just say that this population is the population. We simply have not seen enough of the population, so everything we've seen so far. And we wanted to develop a model that is in between, that is actually modeling people on an individual basis, but we can borrow from the population into those models when we see new patients. And this, thus we created this HDP Flow. This is work in progress. This has not been published yet, probably submitting it, well, not probably, for sure submitting to NeurIPS, whatever it is, the new iteration. But this is the idea. So this is the HDP Flow. It's a generative process. And the idea is that there are latent states, as before, from which the signal comes from. So notice these are not different patients. These are different types of signals. This is one patient, yeah? So you have one state, another state, maybe a third state. Maybe then the patient stays in the exact same state. And as you are propagating, this is generative process, you are understanding the states and you are generating these ideas. So the state distribution changes over time. So you can see the state distribution in the top, whatever left for you. All right, doesn't matter. So then you have this set of states in the end. And the states are not specific shapelets or anything like that. They're distributions from which the new states like that would be drawn. So they are parameters by distributions. And you can do this ad infinitum because this is a non-parametric model. So this is what the model looks like. It is sort of comes from the good old practice of Emily Fox when she was here, and the theory of dirichlet processes that was further developed since she left. But the idea is as follows. You have the data which comes from the states. And the states have various parameters. So the probability over the states, the parameters of the states, the stickiness of the states, how often do we want to transition in and out of the state? All of that is parameterized and learned from the data. And this set of states can grow forever, and you can take different cohorts of data, and you can just keep generating states over time. And it may be that some of the old states will not be useful anymore for new patients, but some of the states will keep coming up, and the new patients can always, the first thing we do is when we see new patients, we can just go to the bank of states and we can say, oh, we already have states that describe this patient. We can start predicting, start understanding these patients right away. And what more is that some of the states will be labeled, because some of the data actually comes with labels from the states. So we can actually use the labels from other studies to label new studies and the patients. This is my dream. We'll see how that works. Yeah. So this is kind of on top, you see the ground truth of the underlying states. You see in the bottom of the estimated states. So what happens is, of course, this tail. This is quite normal for the Dirichlet processes, for those of you who have ever worked with that. There's always this tail of a lot of states that seemingly have been seen in a unique way. We have found some ways to counterbalance that, so we are doing better in that space, but there are some of the kind of standard approaches. This model normally in the past would have been doing sampling, learned by sampling, and this is quite expensive, so we've developed the sort of the black box variational inference to infer the parameters for this type of model, which is significantly faster. And it can generate, of course, a whole bunch because it's a generative model. So it's fantastic, right? We just do AI and somehow the patients benefit, right? No, it's not right. It never works this way, unfortunately. It would have been great. So we actually had several students, including a collaboration with the systems professor, Gennady Pehemenko, who is amazing, to have developed what we call a Lifeline pipeline, where we have developed a full dynamic engine for incorporating the systems, getting them from the database in real time, from the monitors, and doing the inference and streaming it back into to the clinician, to the clinical monitor. So, and what was further interesting is that we actually realized that there is no way for us to evaluate this model offline, because there is no real label of deterioration. The clinicians do not record deterioration in their clinical notes. And so what we have developed, and this paper was just accepted a couple of days ago, what we had to develop is a whole new interface for collecting the labels and intervening on the prediction in real time. So I actually have a video of how it works. So they scan the patient, and then the model keeps running in real time, and the clinician has the ability to drag the prediction, whatever they see, if they disagree with it. So the prediction goes down. The point is that at some point the clinician completely disagrees with a prediction, and they are able to drag it in real time and record what was wrong. They're prompted to record what was wrong with the patient and what was wrong with the prediction, so that we have the provenance of the decisions that are being made, which helps clinicians as well as our model. And so taking this, we keep the evaluated curve and our own predictions, and then we are able to improve our model in a sort of continual learning type of way, which is another project that I'm not going to talk about. So just to give a sense of this this work. It started as a simple cardiac arrest prediction. We had a paper and whatever, 8 months, success. But the reality is that to take it from there and to go in all the way to the clinical practice, it just takes so much more. And I suggest that you work directly with clinicians to actually know what it takes and what is required. So there are a lot of challenges, right? We very often asked to we are very often asked to work with very low frequency events. And in the beginning, I was thinking, okay, it's low frequency, so let's model it as an outlier. But we don't have a class. People who are not having this critical event, they're not also heterogeneous. It's not a manifold of people. They're like, they have their completely different trajectories. And so modeling it and as a sort of proxy variable, making a predictive task and learning a model for a proxy task, and then doing a continual learning on the task that you care about by designing sort of interfaces that will give you the labels that you want, may be the way to go. We also, this interface, I think is in the GitHub, so if anybody wants to use the interface, feel free. Heterogeneity of the population is real, and non-parametric models must be one of the ways to go. I think there is a lot of excitement about deep learning models, which are not really suitable to a lot of the time series tasks as of yet. And I'm hoping that some of these non-parametric Bayesian models that we've been developing are a precursor to maybe some of the more to the models in the future that may be deeper, but I don't think the deep learning models are there yet. They're just not suitable for these types of tasks of time series. And one thing that I haven't talked about is what happens once the model is deployed. So what if it's predicting something that is not, that is off? How do we correct the model? Are we doing continual learning? What improves the model over time? How we come back the feedback loop? All of these questions have to be addressed. So this is it for this project. This was one student's PhD, more or less. And there are many, many other projects that I have that we haven't talked about, from medical imaging to sort of counterfactual modeling in a space, and some reinforcement learning and the churn reduction, and etc. So I'm happy to talk about that. And that's it. Thank you very much for inviting me, and I want to thank my collaborators and my students without whom it would not have been possible. Thank you.
Thank you very much for a really, really great talk. Okay, so for questions, could you come to either one of the microphone or put up your hand, and then I will try to give you the microphone. And also online, you can put in the questions and we will be monitoring them. So maybe I will start with one. You know, like, so I guess in the ICU, you will have like the doctors doing all kinds of interventions as well. So where does that go in? And like, are you, I mean, I'm assuming that is easy to record, so you will be able to kind of use it already when you're looking at your curves. Does that enter in somewhere?
So yes, we've been asking for the data on the interventions for a long time, but interventions are stored in electronic medical records, which are held by some other people. And so it's a lot harder to get all of this data all at once. I'm being recorded, right? So I won't say anything else, but so we don't have intervention data, but we have a project where we can actually detect some of the interventions automatically directly from the HR signal, which is quite nice. So if somebody does a blood test, a blood draw, we can detect it from the signal with basically over 99% accuracy. So we can detect some of the interventions, but not which drugs were used or anything like that. So we the model that we have is able to incorporate that information. Now the question is, are we incorporating it as a sort of additional input feature, or are we using that to create better output labels? So that once we get it, I think we'll have to make a decision about that.
And the doctors just adding it in is just not, they don't have the time at that time to say what was the intervention that they just did.
No, it's really phenomenally difficult that other people are in charge, and so these other people have a different plans and time allocation. I don't know.
Makes sense. Other questions? Happy to give around the microphone as well. Otherwise, I will definitely ask more questions. So, at the end, I think it's like really exciting to, I'll go with one more. So it's really exciting to see how you get doctors into the loop. But how do you motivate them to do it? What is the structure that is set up here so that you can really collaborate with them?
So I early on realized that motivating doctors is a futile idea. So they come to me, and I know they're already motivated, and that's how it works.
Okay.
Yeah.
Please.
Thanks, Anna, for a really great talk. So obviously, like the time series you showed were like continuous measurements, but I'm wondering how you think about incorporating things that are time point. Like you mentioned blood draws, and for example, there's some sort of patient measurements that might be accessed like every few hours, etc. Do you think that your continuous data is somehow like contains some of that information, or that's something that you would add in some way to the modeling of the patient states?
Yeah, so this is an interesting question. We have been working on models like that with, it is sort of an extension of diffusion models, but where we have continuous data, but also the survey data, and we are trying to incorporate both. Generally, the simplest trick, if you just want a response and not to develop a whole new model, we've been basically summarizing some of the continuous data into the chunks that make sense with survey data, right? Because there we were trying to answer different questions. But if you really want to have continuous data and you have every so often some other measurements, the way that we've been doing it is in a sort of mixture of experts type of thing, right? Saying that this is modifying my continuous data, and I'm trying to incorporate the both. So it's not quite an ensemble model, but sort of a continual learning paradigm where we incorporate both.
I have two short questions. So for the time series where you have the probability of cardiac arrest, did you try, so you showed these kind of embeddings, let's say, or latent space. Did you try to throw some kind of disentanglement on this to get maybe a bit better or more interpretable representation?
We did try that initially. There didn't seem to be an advantage of having that constraint in the learning process. It didn't seem to help us much. But yeah, this was part of the project initially. The idea was that we started with a disentanglement.
Okay.
And
okay.
It didn't seem to help.
Okay. And second very short question. Have you tried predicting entire future trends instead of just a probability? And how well does it work, and what's the approach?
Like a seek-to-seek type of model where you're using the loss of the prediction. Yeah, so the fit model, in fact, if you look at it under the hood, we are trying to predict the sequence and then trying to compare the two sequences and see how different they are. And in that case, it works really, really well. I think it always, as with all predictive models, it really matters what your ultimate goal is, right? With the representational learning, it wasn't really that we were trying to predict the future sequence. With a cardiac arrest outcome, it wasn't really the sequence that mattered. It was whether the event has happened or not. So we did do that in the FIT model. That was part of the generative model, and there was a time series model which made a prediction about what sort of the counterfactual outcome would be if the patient wasn't getting worse.
Okay, interesting. So why I was also asking this, so we also work a lot with medical people, medical doctors, and for us it was that if you just give a score, they were a bit hesitant. They wanted kind of what you said all along, like the features that really matter, the biases and so on. But when we gave them kind of like a future prediction where they can kind of reason about, okay, this, let's say here, the trend that we see, or the, let's say, Chaplet or whatever, they could reason themselves and they were more happy. So
yeah, that's I think that's interesting, and you just have to do it on a case-by-case basis. We do that in another project that we have in the emergency department, where our goal is to predict the sort of the patient census, how many patients will be there for the next 24 hours, and so that's the whole goal, is to predict the sequence of the numbers of patients in a unit over the next 24 hours. So we definitely are aware of this prediction task. It's just, again, these are constant discussions with the clinicians about what they want and what they feel comfortable with.
okay,
Yeah.
thank you.
But yeah, I hear you.
Yeah, thank you for this very inspiring talk. One quick question: how transferable is this model to other ICUs and clinics? So how important are the input features, and what happens if in another hospital ICU this specific feature is not tracked or monitored in a patient? Can this model still be applied? That's one question I have, and the other goes maybe but in the direction of what has been asked before, is do you think it will be possible also with these kind of models to at one point track this down in more detail, to basically say if this continues to happen in the patient, then we can predict or say that in 4 hours or 3 hours this event will happen, or even say if you will be able to attenuate this specific signal or problem in the patient, such as the blood pressure, the risk for cardiac arrest will also drop by this and this %.
Okay, so your first question, we definitely want it to be used everywhere, right? That's the goal. I have several of my graduating PhD students working on sort of transferability, and I think what we are finding now is that you can do pre-training, right? Use this model as pre-training, but you do have to specialize it per hospital, per place. If you don't have the feature or multiple features present that you're monitoring, then there's always a way to mask and not use that feature at all, depending on how its importance towards the prediction. But oftentimes, people, what I've been seeing is you have to retrain the model, right? You just take the structure and the architecture and you retrain it for your local model, for your local hospital, especially in the physiological space. There's always a lot of data. If the data is being stored at all, it is very quick to accumulate a lot of physiological data. So I would say, yes, by all means, but retraining might be more useful than just specializing the model in a particular place. Now, what you are saying, so predicting the sequence by saying, okay, the blood pressure will keep going up if, something should be done to the blood pressure. I think that's an important question that we didn't necessarily, the model as it is not necessarily addressing right now, right? So this this is something that I guess we will have to discuss with our clinical collaborators and say, well, is this something that you would find useful in your setting, in your department? And I think it's a very, I mean, I don't see why they would say no, but it sort of builds on one step at a time in a collaboration in terms of what they need and us trying to build the best possible tool for that. But I think both of you are right. This is an important way to give a sense of the prediction. And the emergency department exactly asked for that. So,
Cool, thanks.
Sure.
Thank you very much. It was a wonderful talk. So I was just wondering, when you're selecting this feature, and instead of like choosing this 50 top features, which has like a top this I function values, can you formulate and like in the setup of like the multiple testing and use FDR or something like that?
For
Like,
what?
I suppose like when you like look at this I function, you're doing some sort of like looking for like independence or conditional independence. Maybe you can do testing and like and select the features which probably like with the lowest p-values.
You mean, why is the question, why don't we look at feature association by just
So it was
doing the
just like
correlation
the
peak
so
and by minimum
50s,
p-val?
like the rationale behind like choosing the 50 top 50 like features which like the highest I values. It's not like, I mean, it's ad hoc, right? Like instead of that, can you do some more?
We're not picking features by the
Oh,
high
it's not
values.
like, oh,
No.
then I don't know. Say,
No.
I was just okay. So sorry.
Yeah, there is the whole score with a difference between the distributions of the features prediction and observation to make sure that the feature, the subset of features, actually not individual feature, but subset of features actually have an input into that loss between the KL divergences between what we expect versus what we observe.
And the number of like subset, like candidates in the subset is like fixed or vary?
No, it's varying. It's for
varying.
all subsets.
Okay.
Yeah.
When you condition on XX, you need to compute that score for the subsets. Do you need to compute that for all the potential subsets, because that's an exponential number of subsets?
Yeah. So that's right. But if you think of the time series, usually I think we had maybe 6 or 8 signals.
Okay.
Right. So
I see.
it's very different. It's not like you have a, the set is a full set of genes. You really have very small number of time series features, right?
So
Usually it's
affordable to compute it for all
subsets. Yeah. And you can pre-store and update.
I see. Thank you.
Yeah.
For the KL divergence, do you also use some like parametric model like Gaussian, where you can compute it in closed form, or do you do some other approximations for those?
We did a bit of, I think there was a bit of sampling for the X given, for Y given XT minus 1 XS. There was sampling that we did. Yeah.
Thank you.
Great. Okay. Thank you very much. Since we're over time, Manolis, you'll ask directly. And then, yeah, I know that. Thank you very much for a really wonderful talk. Thanks.